Welcome back. So today we're going to start talking about genetics. So while we have discussed genetics before, or I should say that it's come up in conversation before in my lectures before where I've discussed genes and DNA, now we're really going to dive into it. So as much as we've talked about chromosomes and talking about mitosis and meiosis, now it might actually start coming to life as we start to talk about both Mendelian and non-Mendelian genetics or genetics as a whole. So something that you want to understand is that when we talk about genetics, we're really talking about passing on information, passing on really traits from one generation to the next. So your mother and your father, they contributed to your genetic makeup. Fish odor syndrome, where individuals smell like rotting fish, this is an inherited disorder. So for whatever reason, this textbook likes to open up talking about how this is a trait that can be passed on from parents to offspring. So humans have 23 pairs of chromosomes, and we looked at that when we talked about mitosis and meiosis. You get 23 from your mother and 23 from your father. You have your maternal copies, your paternal copies, and that is going to make up 46 chromosomes. But remember, we get 2 number 1s and 2 number 2s and 2 number 3s all the way up to 22. And then you get an X and an X, or an X and a Y. So with any chromosome, it's going to be made up of segments, and those segments are made up. Those segments are known as genes. So DNA or chromosomes are made up of these long strands of DNA. And when we look at a section or a segment of that DNA or of that chromosome, we're going to call it a gene. A gene is something that is going to code for a particular trait. So for any gene, you're going to have 2 versions of it. You're going to have one from your mother and one from your father. So if you look at your parents, you're going to see that, hey, if this is, we could say this is, chromosome 1, and this is also chromosome 1 of your mother's. And then your father had 2 chromosome 1s as well. So this is chromosome 1. This is one of his chromosome 1s, and this is another of his chromosome 1s. Now, they're going to pass on 1. So let's say this one gets passed on to you, and this one gets passed on to you, and they each have a particular gene. Now, these genes can come in different versions, and different versions of genes are going to be known as alleles. So different alleles are going to code for different traits, or they're going to create different looking things or different traits, characteristics. So if you get one from your mother, all right, and one from your father, that's what you're going to end up with, and you're going to have this. Now, when we look at this gene, you get one allele or one version of that gene from your mother and one allele or one version of that gene from your father. Now, in meiosis, it produces human gametes with just one copy of each chromosome, and that's how we have our sperm and egg, and that's how your mother can have 46 chromosomes and your father can have 46 chromosomes, but they're only contributing 23 to you, each of them, and now that's when you come together and you also have 46. So the child inherits one set of chromosomes from each parent, and thus 2 copies of each gene or alleles. So when you look at something like fish odor syndrome, just as an example, we look at these different alleles. So this is the same gene or the same segment of DNA on both of them. But you see in this one, you have the normal allele for the FMO3 gene, or the gene that is going to dictate if you have fish odor syndrome or not. So you have the normal one, which means you don't have fish odor syndrome, and then you also have the defective one, which means, hey, you do have the allele for fish odor syndrome. So what we see is that this is a recessive trait. Now, whenever we're talking about dominant recessive, we typically see that there are 3 options that you can get. So you could even, if we wanted to look at this in terms of a Punnett square, so a lot of times people remember from high school that they learned about Punnett squares. and it was set up where there's an uppercase and a lowercase letter. So uppercase letters represented our dominant alleles, and lowercase letters represented our recessive alleles. So if we were going to look at this in that way, we would say that this one, our normal one, could be our uppercase A, our dominant, and this one could be our lowercase a, our recessive. Now the father might have the same thing where he has one recessive allele and one dominant allele. Now, when we look at this, we see that, you know what, if they have at least one dominant allele for this, then they are not going to have the trait because the trait is recessive and the dominant trait is going to overpower it. So both of these individuals are what we would consider normal, or they don't have the fish odor syndrome, but they're known as carriers or heterozygotes because although their traits don't show it, they have this lowercase letter hidden by the uppercase letter. All right. So by looking at this, we say, hey, these two individuals don't have it, but they could create a child that has it, because if the child gets this recessive allele and gets this recessive allele, okay, these create the individual, then this individual has two recessive alleles and it doesn't have the dominant allele or an uppercase letter to kind of hide or mask this recessive allele. So you have people that are uppercase that we refer to as homozygous. Homo meaning the same. Homozygous dominant. All right, meaning they have two dominant ones. Then you have heterozygous, meaning they are hetero, meaning different. Heterozygous. And then you also have lowercase, which is going to be, I'll write this down below, is going to be known as homozygous recessive. And whatever the trait may be, if they are dominant, they're going to show that dominant trait. If they are heterozygous, they're going to show that dominant trait. And the only time you see a recessive trait, you don't see it in this because it's masked by the uppercase. But in this, they are going to be a, lowercase a, and that there is nothing to mask it. That recessive trait is going to be shown. So they show you how you have these two heterozygotes, because they're uppercase a, lowercase a, or known as carriers. They are contributing, because remember, when we talked about meiosis, everything's getting split up, and you can pass on, she can pass on either an egg that has this in it or an egg that has this in it, and he can pass on either a sperm that has this in it or sperm that has this in it. So by chance, if a sperm with the recessive allele and an egg with the recessive allele come together, it's going to make this homozygous recessive organism that is going to show that fish odor syndrome. So when we like to make Punnett squares, we're going to look at them and we'll say, hey, you know what? The father has uppercase a, lowercase a. The mother has uppercase a, lowercase a. And now when we put them together, these are the options. You can either make a child that's uppercase a. You can make one that's uppercase, lowercase, another one uppercase, lowercase, and then a lowercase. So since they are heterozygous, meaning uppercase, lowercase, they have a 25% chance of being of the offspring being homozygous dominant. You have a 50% chance, because 25 plus 25, that they are heterozygous. And then you have this 25% chance of being homozygous recessive, which means they have this fish odor syndrome. All right, so let's keep looking at this. Now, heredity is the passing of characteristics from parent to offspring through the parent's genes. So since genes are going to dictate what your traits are, or what you look like, or what your characteristics are, you're passing on not the traits necessarily, but the genes that are the instructions for the traits. So when you look at, some offspring or some children, they look very much like their parents. All right. because they have similar genes, or they have the same genes. So some things that point to this, which make it very obvious, are things like selective breeding, or sometimes called artificial selection. So as much as we'll talk about natural selection when we get into talking about evolution, we like to talk about artificial selection. So in natural selection, nature is choosing which traits will survive, which organisms will live long enough to reproduce and pass on those traits. With artificial selection, it's humans doing the selecting. We're artificially selecting which organisms are going to live and reproduce and pass on those traits and make more of them with more organisms with that trait, and which organisms are not going to be passing on traits or reproducing. So once breeders recognize the existence of heredity, selective breeding, such as for reduced size in horses, also you think about dogs, people want these little teacup dogs. That's another example. People want big bully breed dogs, very strong. That's another example where if you look at pit bulls, people want these big bully breeds. Well, they're going to take the biggest mother or the biggest female from a litter and the biggest male from another litter, and they're going to mate them and try to make a really big one. And then when that one reproduces the offspring, we're going to pick a really another big one. And over many generations of selecting, artificially selecting or selective breeding, you're going to see that you're changing the traits. You're really weeding out the traits you don't want, and you're seeing more and more of the traits that you do want. Now, the easiest example is when we talk about things like single gene traits. Now, there are many traits that are based on multiple genes, and multiple genes are going to influence what we see, what trait we see, what we consider the phenotype to be. So we have the genotype, which is typically we're going to say it's either going to be AA, or lowercase a. And then you have the phenotype, which, if we think about our fish odor syndrome, it's either going to be fish odor syndrome or no fish odor syndrome. So now when we look at single gene traits, some traits are determined by the instructions an organism carries on just one gene. Most human characteristics are influenced by multiple genes in the environment, but when we start talking about things like Mendelian genetics, we like to open up just looking at things that are a single gene that is going to dictate what the trait is. So fur length in cats, coat color in cats, these things are based on a single gene. What gene did you get? That one gene. What allele did you get for that one gene is going to dictate your coat color. Are you white? Are you gray? Are you black? And then for length, also, are you long hair, short hair, based on one gene. So now Gregor Mendel, he learned about heredity by conducting experiments. Previous to the mid-1800s, they didn't understand this concept of heredity, how there could be recessive traits, and the recessive traits might not be seen in the parents, but they can be passed on. You could see in future generations. So why do parents give their offspring what do they give their offspring that confers similarity? Now, once upon a time, they thought that, you had these animalcules or these, miniature humans that were cells that they grew up to be adults. But now we see that sperm is just genetic information. It's just that DNA in there as well as egg. But it wasn't for a long time until we figured this out. Now, with Gregor Mendel in the mid-1800s, he was able to do this by conducting experiments, and that's any good science is based on research and experimentation. So he took these pea plants that he had, and he started to explore genetics, what we consider Mendelian genetics. And when we move forward and we talk about kind of the nuances or the more complex ideas, we can call them non-Mendelian genetics. But pea plants are an ideal study organism because they're easy to maintain, easy to breed, and they reproduce very quickly. So you wouldn't want to do experiments like this, especially early on in the mid-1800s, you wouldn't want to do experiments like this on an elephant. An elephant lives a very long time. They only reproduce so many times in their life. It takes them about 2 years for them to have a child or have an offspring. So that's not a good example. But pea plants make it very easy. So what he did was he looked at a number of traits. Just for example, flower color, pea shape, pea color, all things he looked at. And there were different varieties. You see that you had different variants or variations. You had purple flower versus white flower. You had round peas versus wrinkled peas. You had green peas versus yellow peas. Other traits, pod color, pod shape, flower position, plant height, all of these things were researched. Now, what he did was he bred them. So he used true breeding plants, meaning plants that were that no matter what, as long as you continue to mix them together, they made the same plant. So he took purple ones, which no matter how many times you mix a purple with a purple, it's always going to make a purple. And he took white ones, which no matter what, as long as you mix a white with a white, it's going to make white. So he called these true breeding, because as long as you didn't do any mixing, they were going to come out the same 100% of the time. Purple with purple always makes purple, and white with white always makes white. So he was able to prove that these are true breeding colors. Now, he conducted these studies, and it helped him to understand heredity and how things are passed on and how genes are going to influence what trait is going to be shown. And he focused on easily observed and categorized traits in garden peas, and he applied this methodical experimentation and rigorous hypothesis testing to determine how traits really are inherited. Now, he discusses segregation. So you've got 2 copies of each gene, but put only 1 copy in each sperm or egg. So while he didn't know this, we now know that is segregation, where you have a sperm and an egg, and when you're doing meiosis, you're going to have 2 copies, but you're only going to put 1 copy. You're only going to put 1 chromosome 1, 1 chromosome 2, 1 chromosome 3 in a sperm or an egg. So you have a dominant trait or dominant allele, which is going to mask the effect of the recessive trait. Remember, the traits aren't the actual gene themselves. It's the gene is dictating how the trait will be. So Mendel identified the purple flower trait as dominant and the white flower trait as recessive. So what he did was he mixed a purple with a white, and he saw a purple always come out. So we know that if you mix a purple with a purple, it always makes purple. If you mix a white with a white, it always makes white. But if you mix a purple with a white, it's always going to make purple. But he said that if he then were to cross a new purple that came from this purple-white mix with another purple that came from this purple-white mix, you ended up with some of them coming out white. So this is where we see our Punnett square, where some of them are homozygous dominant, some of these might be heterozygous because they would just look purple. Both homozygous dominant and heterozygous are both going to come out as purple because they're both going to have at least 1 dominant allele. And then you're going to have ones that are homozygous recessive, and they're going to come out white. Now, 3 important ideas from Mendel's research. Each parent puts a single set of genes into each sperm or egg it makes. Offspring receive 2 copies of the instruction because you're getting 1 copy from mom, 1 copy from dad. And the observed trait in an individual depends on the 2 copies of the gene it inherits from the parents, and which ones are going to be recessive, which ones are going to be dominant. So his law of segregation, while he didn't understand DNA yet, we didn't have a knowledge of what DNA was and how it specifically worked, he had this idea of you're going to have these traits, and they're going to be dominant or recessive, and you're going to pass them on. So, according to Mendel's law of segregation, only one of the 2 alleles for a gene is put into a gamete, and at fertilization, where the sperm goes into the egg, the offspring receive from each parent 1 allele for each gene. So here we saw after he mixed a purple with a white, everything came out purple. But when you mix the purples that came from them, you're going to end up seeing that, hey, Some of them came out homozygous recessive, some came out meaning white, some came out heterozygous, and some came out homozygous dominant. So this actually, since it has one purple, which is the dominant, and one purple, these have two purples, these were all purple. But this one didn't have any purple to mask it, any dominant, so the recessive trait, the white, was shown. So now each parent puts a single set of instructions for building a particular trait into every sperm and egg that he makes, and these instructions are called genes, and different types of, or different versions of that gene are referred to as alleles. Now, phenotype is going to be the trait itself. Okay, so far we've been looking at genotype versus phenotype, where genotype is either going to be AA, or AA, either going to be uppercase, 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 lowercase, or lowercase, lowercase. But our phenotype, so this is our genotype. Oh, this too. All three of them are our genotype. Our phenotype is either going to be has the trait or does not have the trait. So you have two, or if it's something else, it could be is purple, is white. Those are two options. So the genotype is really the setup of the genes, and the phenotype is what we see. So something like albinism, you're going to see in a Punnett square. So albinism is a recessive trait. So this albino giraffe is lowercase m, and the letters don't really matter. It's just a representation. So we could say letter A, letter M. In this example, I like to say they used to like to use M's. So then the father is actually homozygous, which means uppercase M, uppercase M. So they took a true breeding homozygous dominant giraffe and bred it with a homozygous recessive, which is lowercase m for albinism. And what you got was that you have this one that has uppercase, lowercase, this one has uppercase, lowercase, then this one is uppercase, lowercase, and this one is uppercase, lowercase. So all of them are going to look like the father because they get this uppercase from the father, and it masks whatever else they get from the mother. Now, when you cross a mother with a heterozygous father, where he's uppercase, lowercase, well, now he has an uppercase, and she has an uppercase. So when you cross a heterozygous mother with a heterozygous father, they're each going to have an uppercase, so you're going to have a homozygous dominant. And they're also going to have a lowercase, so you're going to have these 50% chance of a heterozygous. But you also have these two that are hidden. So while both parents are not albinos, they carry that allele for albinism, and you can have a quarter of your offspring. If they were to have 4 offspring, one of them, most likely one of them, just based on odds, will be an albino. Now, segregation and fertilization are chance events. So it's not like, oh, if I have 4 kids, it's definitely going to be the, 3 of them will be this way and 1 of them will be this way. It's a chance event each time. So it really depends on which sperm and which egg are going to come together. The probability of certain genetic outcomes can be predicted. So that's when we look at the chances. So something like the pigmentation or the coat coloring, you see that it's 100% chance of having this albinism, and you have a 50% chance of having the albinism from the father. So you multiply them together, sometimes referred to as the product rule, and it means that there's a 50% chance that the offspring will be albino based on what the parents are. Here's another example, 50% chance of having Tay-Sachs. Father, also 50% chance of having Tay-Sachs. So while both of them do not have Tay-Sachs, they are both carriers, and the child has a 25% chance of having Tay-Sachs disease. Now, a test cross enables us to figure out which alleles are an individual carries. So with something like alligators being albino. Well, we know what the albino alligators are, but we don't necessarily know what the colored alligators are. We don't know if they're homozygous dominant or if they're heterozygous. So you can take one and you can say, all right, I know that the albinism or the white color is going to be homozygous recessive, which means lowercase m, so I can input them. And then I know that the other organism that has color is either going to be uppercase M, or they could be uppercase M, lowercase m. So when we work this out, we would see that, hey, in this scenario where we know that the uppercase M that they definitely have, because they at least, the first one's at least uppercase M, is used, we see that they're going to be, this alligator is going to have normal pigmentation, this one too. But then if we have one that is going to have a second uppercase M, it's going to make two more, meaning 100% of the time, if they are heterozygous dominant, 100% of the time, it will be colored. The mix of this white with this one, if they are heterozygous dominant. Homozygous dominant. If they're heterozygous, then we should see that 50% of the time, you're going to have these colored ones. The other half of the time, you're going to have these albinos. So if we were to cross these, and we took all the eggs from these alligators, and we counted up what the offspring looked like, we would either see that they are all heterozygous organisms, they all are pigmented, or they have coloring, and we would know, okay, if that's the case, the father must have been homozygous dominant, must have had been uppercase M, uppercase M. But if we see that half of the offspring are colored and half are not colored or albino, then we would know that the father must have been heterozygous, meaning uppercase, lowercase. So in a test cross, an individual that exhibits a dominant trait but has an unknown genotype, meaning we don't know if it's heterozygous or if it's homozygous dominant, is mated with an individual that's homozygous recessive, because homozygous recessive people or organisms are always going to be lowercase. So we know what they are, because if it shows that, that means they do not have a dominant allele. But we don't know on the dominant ones, the ones that show the dominant trait, if they're heterozygous or if they're homozygous dominant. So the phenotypes of the offspring reveal whether the unknown genotype individual is homozygous dominant or if they are heterozygous. So sometimes we like to organize these into pedigrees, and a pedigree is a way of showing the movement of traits through a family. So you have these basically a diagram that shows you which organisms had a trait, which organisms did not have a trait. In this one, we're looking at just an idea of a trait, and anything that's shaded in is going to be have the trait. Anything that is not shaded in is not going to have the trait. And we see that anything that is a square is going to represent a male, and anything that is a circle is going to represent a female. So Grandpa had it. He mated with Grandma, and Grandma, and when Grandma had the babies, they created a daughter that had it, a son that didn't, and another son that did have it. Okay, these are spouses. So when you see this connection below, those are going to be spouses. This is a spouse. And this child right here, from this first set, they married someone from this other set. So you have a grandfather that didn't have it, a grandmother that didn't have it, and all of their children did not have it. But now you see this gentleman that had it and this female that had it. They mated, or they reproduced, and they created these offspring that a daughter didn't have it, a son had it, or didn't have it, and a daughter did have it. So now, with something like anury, which is no tail for things like dogs, if a dog has no tail, but both parents do, then we know with certainty each parent's genotype. So we know if anury is a recessive trait, then if both parents didn't have it, they must have been heterozygous. and the offspring must have received the 2 recessive ones. All right, this one, once again, could be heterozygous dominant, homozygous dominant, or heterozygous. And then this organism is lowercase, lowercase. So what are the options? Well, we would have to work out the Punnett squares, where we would see that, okay, the mother is either going to be uppercase, uppercase, or is going to be uppercase, lowercase, and we know that the father's lowercase, 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 lowercase. So there's a number of options that could come out. Now, it's not always as simple as having purple plant or purple flowers or having white flowers. Sometimes we're going to have incomplete dominance, which is the phenotype of a heterozygote, meaning uppercase, lowercase. So if you're going to have uppercase, lowercase, or uppercase, lowercase, whichever letters you want to use, the phenotype of a heterozygous is the intermediate between the phenotypes of the 2 homozygous individual or homozygotes. So if you look at these flowers, you have these flowers that show incomplete dominance, where the ones that have you have 2 white traits, that's going to make white ones. You have 2 red traits, that's going to make red ones. But when you mix the white with the red, it doesn't just make ones that are either red or white, like we see with the pea plants. It actually makes ones that are mixed. So if you have one of each, it's going to make pink, and that's incomplete dominance. So now, when you're mixing these different ones, you can end up with a pink, a red, or a white. It's not just purple or white. So it really depends on what type of traits we're talking about. In the pea plants, we saw that it was one or the other. They showed complete dominance. But with other plants or other organisms, they can show incomplete dominance. There's also codominance, where we see a blending. So heterozygous individuals show features of both alleles. I shouldn't say blending. Blending is more incomplete dominance. Codominance is having both. Okay, so they show you this example of, hey, if the father is wearing stripes like this and the mother's wearing stripes like this, he kind of has a plaid thing going on, or checkered thing going on. So sometimes the effects of both alleles in a heterozygous genotype are evident in the phenotype for that trait. If they are showing incomplete dominance or codominance. With incomplete dominance, you see that they are kind of an intermediate or a blending of the two. With codominance, you're going to see that both are shown. With blood types, this is important because you see that someone can have type A, type B, type AB, or type O, and it really depends on which alleles you are going to have. So if you get an A from your mother and a type A blood gene or allele from your father, you're going to be type A. If you get an A and an O, you're going to be type A. If you get a B and B, you're going to be type B. If you get a B and an O, you're going to be type B. If you get an A and a B, you're going to be type AB. And if you get an O and an O, that's the only way you're going to be an O. So antigens are like signposts in the body's immune system, or like flags that we raise. And antibodies are immune system molecules in the bloodstream that attack foreign invaders. So we want these antigens in order to kind of flag the cells that are ours, and us to be able to recognize the cells that are not ours. So we see things like the ABO system. We also see positive or negative. That's the RH factor, or RH markers. A single gene with 2 alleles determines the presence of RH marker, not an example of multiple allelism. But individuals with 1 or 2 copies of the dominant RH cell marker are going to be RH positive, and the ones that don't have any are going to be, or they have their recessive allele, they're going to be RH negative. Now, multi-gene traits, how are continuously varying traits such as height influenced by genes? How do people have, with people, it's not like you're either 5 foot or you're 7 foot. There's so many variations in between, and we consider these polygenic traits, and they're influenced by many different genes. Skin color is really dictated by 9+ genes. So, when you look at skin color, it's not like you're just white or black or white, black, Hispanic. There's so many variations. White people, there's so many different shades of white people. Some people are very pale. My wife is extremely pale, and then my brother has kind of an olive skin, and then you see other black people where some are very dark skinned, some people are very light skinned. This is because it's all based on a number of genes that are going to kind of create this difference in really how light or how dark an individual is. When the effects of multiple genes contribute to the ultimate phenotype, it is described as an additive effect. Now, autism is a development disorder that seems to be the result of alterations in numerous genes. So, possibly 10, 20, or even more genes are involved. And what they're seeing now is that it's also influenced by the environment, which they consider this to be epigenetics, looking at the way that the environment is going to turn on or turn off the genes. So we can turn on genes, we can turn off genes, and that's going to also influence them. So the symptoms and the intensity of symptoms of autism varies widely from one individual to the next. And sometimes you'll hear this term, the geek theory of autism. So many traits, including continuously varying traits such as height, eye color, skin color, they're going to be influenced by multiple genes. Now, some, are going to be just one gene, and it can actually influence multiple traits. So you see something like sickle cell anemia, it's just one gene. So you can have an allele for no sickle cell anemia or an allele for having sickle cell anemia. So people that are homozygotes or homozygous dominant, they produce no sickle cell. They don't have the sickle cell allele, either of them, because you're getting one from your mother, one from your father. You have heterozygotes, which are going to have one allele for it, and the other allele they get from the other parent is not going to have it, which is actually helpful in creating immunity for malaria. So it's beneficial in some areas, some countries that are stricken with malaria to be heterozygotes. But then you have people that are homozygous recessive or homozygous for having that allele, that the sickle cell allele, and it helps them to also be immune to malaria, but it gives them a lot of health conditions where they're going to have trouble breathing, hypoxia, and that's where you hear about people suffering from sickle cell anemia. As much as it's good for fighting for staying safe from malaria, it is not necessarily beneficial to be homozygous for having the allele on both of the genes that you inherit. So pleiotropi occurs to when gene influences multiple unrelated traits, like the trait for having sickled cell and the trait for being able to do well when met with malaria. So in pleiotropy, one gene influences multiple unrelated genes. Most, if not all, genes may be pleiotropic. All right, so there's a lot to it. It's not as simple as just looking at Mendelian genetics or complete dominance, where we just see setting up a Punnett square, and you're either going to have purple or white flowers. Not that simple. There's a lot more to it. So keep these things in mind as we move forward and we continue to talk about other factors that play into genetics, as well as talking about evolution and how the genes are going to dictate which traits you are, and how your traits are going to ultimately interact with your environment and dictate if you are fit for your environment, or if you're going to do well in your environment, or you're not going to do well in your environment. You're not going to be able to live long enough to reproduce, pass on those traits, and make more organisms. So keep these things in mind as we move forward.